We will now load our serum and cell lysate samples in our gel. And then we will be running the gel electrophoresis process. But first, we need to load our protein ladder. So this will be our basis for the protein later, the bands. We need to load 5 microliters of the protein ladder into well number 1 of our polyacrylamide gel. So we poke the tip of our micropipette at well number 1 and slowly push the plunger, avoiding bubbles along the way. Next, we will load our serum sample, this time in well number 3. And then instead of 5 microliters, we will be using 30 microliters of this serum sample. So keeping the tip steady with an aim, and push the plunger and expel all of the samples without forming bubbles. Then we will load the cell lysate sample in well number 5, 30 microliters of it. Making sure that there's no gap between the tip and the sample, we then insert the tip and push the plunger down. You notice here there's a bit of a mistake. Some of the samples went into wells 4 and 6, but that will be fine. Then we'll finish setting up. We'll cover the tank with the correct orientation of the electrodes. And then we insert the other end of the wire to the voltage regulator. And then we set the voltage to 200 volts. And the current will be at 400 amperes. And this will be run for 40 minutes. So you would know that it's running because there will be visible signs, especially at the anode, the positive portion or side. You can see the evolution or the formation of bubbles as the electric current flows through the wire. And here you can see the migration of our samples down our polyacrylamide gel towards the anode. Notice how there are some mistakes here for wells 4 and 6 and also 2, but that will be fine.